Hi everyone. One of the reasons I became a biologist is that I really like to know how things work. Understanding ecology has allowed me to see forests and rivers in a different light. Understanding Darwinian theory has made me appreciate the common ancestry of life on Earth and the ways in which natural selection has edited variation in populations so that over time, organisms have become better adapted to their environment. Understanding the ways in which cells work has forced me, a big picture guy, to look for and appreciate the world we cannot see with our naked eyes. The modern synthesis of evolutionary theory, led by eminent scientists like Ernst Mayr, was an important step in squaring evolution and genetics, postulating that the entire genome is the target of selection, which suggested that selection could actually work to facilitate the coordination of genes and the regulation of multiple genes at once. This week, we are going to dive deeper into a couple of genes, or rather groups of genes, and learn how they are regulated. Okay, so to get started in understanding, some of the ways in which genes and the expression of genes are regulated, I'd like to start by talking about this specifically in prokaryotes. Now, as you probably remember from Bio 104, genes code for proteins. Okay, they contain the code and the proteins that are translated from the genes express phenotypes and do the work of cells. Some proteins, for example, act as enzymes, which catalyze chemical reactions in the body, such as the biosynthesis of stuff we or our cell needs. Now, again, this is review, but the genes themselves are located in the nucleus, and they're regions of chromosomes or DNA molecules. And when it comes time to translate the genes into protein, the DNA molecule itself sort of unwinds in the region that needs to be translated, but there's not a direct translation between DNA and proteins. There's an intermediary molecule called the mRNA, or the messenger RNA, and this is the part of the nucleotide sequence that contains the code that will actually be translated outside of the nucleus. Now, as you know, I like to connect. I think that discussing these concepts through the lens of Darwinian theory is always helpful. And so let's not forget that Darwinian theory predicts that genes will be expressed in a way that maximizes the use of limited resources, such as amino acids or other biosynthetic building blocks. Now, this means that their expression must be regulated. And there are several ways this regulation works that I'd like to talk about over the next few minutes. First of all, and this is one that we talked about in Bio 104, but there's feedback inhibition, where the product of a reaction itself it directly interferes with the process of making that product. And so we'll look at this with an example in E. coli in a little bit, but as the product builds up in the cell, this feedback inhibition can prevent the overproduction of the product itself. There are other ways, and this is sort of the focus of this video and the lecture that we'll have in class. There are two categories of gene regulation. First of all, there's negative gene regulation, and we'll look at how some genes or sequence of genes are repressible, and the example that we'll use is the tryptophan operon. And then in lecture, we'll talk about an inducible operon, such as the lack operon, and how this works. And then I'll briefly cover positive gene regulation at the same time. I'd like to take this this opportunity now to make another connection to a previous class. In Bio 105, when we talked about predator-prey, and when we talked about sort of exploitative relationships in general, such as herbivory or parasitism, we framed this in this body of theory called optimal foraging theory. And remember that optimal foraging theory predicts that resources will be exploited in a way that maximizes energy gain. A predator, for example, will maybe ignore or avoid very small prey items in favor of medium or larger prey items, which have a higher return on investment. There's an investment in foraging, right? Similarly, organisms will prioritize foods which are more nutritious and or take less energy to harvest. We'll connect this with our exploration of gene regulation in E. coli when we talk about the lack operon and how different environmental conditions inside the cell will make it more or less likely that those genes associated with the lack operon are activated. Now, I've used a word that you might not be familiar with a couple times now, and that word is operon. Here you can see a diagram of a segment of the E. coli genome that's involved with the synthesis of tryptophan. Tryptophan is something that E. coli needs, and so if it's not provided with it, it has to synthesize it. And the synthesis is, of course, facilitated by cell products that originate in the as genetic code. So what I would like you to do now is pause the video and take a look in your book or online and jot down the definition of the term operon. Okay, so when I look up the term operon in the textbook, this is what I get. It's a unit of genetic function found in prokaryotic organisms consisting of a promoter, an operator, and a regulated cluster of genes whose products function in a common pathway. Let's take a look at one of the operons that are featured in your textbook, the tryptophan or trip operon. Now, E. coli synthesizes the essential amino acid tryptophan when it's not provided in the medium in a multi-step pathway. Each reaction in this pathway is catalyzed by a series of 5 enzymes, and they are clustered. The genes that code for those enzymes are clustered together on the same chromosome. This is what an operon is. Like we'll see with the lactose operon, a single promoter serves all 5 genes. Now recall from Bio 104 that a promoter is a site where RNA polymerase can bind to DNA and begin transcription. The cell can translate this one mRNA molecule transcribed from the DNA into 5 separate polypeptides because there are start and stop codons marking the beginning and end of each individual gene. Now, we'll go into more detail about the various units in the tryptophan operon. For now, I'd like you to pause the video and take some time to think about the advantage of clustering genes of related function into a single transcription unit with a single on-off switch. Be prepared to discuss this in class. Okay, Let's talk about regulation of the tryptophan operon. We learned in Bio 104 about feedback inhibition, where the product of a reaction can act to inhibit the reaction itself in order to minimize the overproduction of the product. This saves the cell energy and conserves valuable resources. You can see from this diagram that tryptophan does this. It acts in this way on reactions that result in tryptophan synthesis by inhibiting the activity of the first enzyme in the pathway. This tends to be a rapid response. But this is not the only way that tryptophan inhibits the production of tryptophan. It can also work by regulating the expression of the genes in the operon itself. To understand this, we need to dive deeper into the structure of the operon and the associated genes. As you can see in the diagram, in addition to the genes of the operon contains a promoter site, which I've already mentioned. This is the site where RNA polymerase binds to the operon itself and enables the beginning of the process of transcription. But there's another region of the operon within the promoter region called the operator, which functions as an on-off switch. The operator, as you can see, is positioned within the promoter region of the operon and controls access of RNA polymerase to the genes. The operator contains a binding site for a repressor protein. In this case, it's called the tryptophan repressor, or the TRIP repressor. The TRIP repressor is a product of a regulatory gene located outside of the operon called TRIPR. In the case of the TRIP operon, which is said to be repressible because transcription is usually turned on but can be inhibited, the repressor is normally inactive and does not bind to the operator region. Again, this means that transcription is kept on. In order to turn it off, the repressor protein has to be activated. And guess what's the activator of the repressor protein? It's tryptophan. Tryptophan in this case is called a co-repressor. And so in the presence of tryptophan, this repressor protein binds with the tryptophan, and it physically changes the shape of the protein to activate it, and it makes it so it's possible to bind in the operator site, effectively blocking off RNA polymerase's access to the genes that code for the enzyme. This means the transcription process is inhibited. Well, thanks for your attention. This is about all the time we have. For class, as I mentioned, we're going to be talking about another operon that's in the E. coli genome. It's the lactose or lac operon. But before we do that, I'll open up the space for questions or discussion. And in particular, I would like you to consider the following questions. Be prepared to discuss these. For the first one is one that I already mentioned. What is the advantage of clustering genes of related functions into a single transcription unit with a single on-off switch? I think that this is an important question that we address together. The second question is, why does it make sense for the tryptophan operon to be turned on by default? Why does it make sense for it to be repressible? The third question is, can you imagine a situation where it would make sense for an operon to be turned off by default, but to be inducible? So again, thank you for your attention. I look forward to seeing you in class and in lab this week. Bye-bye.